Well, I will start without further ado. So as Netta very kindly introduced me, I really am interested in how natural populations evolve. And my research has taken me to studying a whole bunch of different systems that range from Drosophila melanogaster and malaria, dogs, New York City rats, and most recently my interests have been on the human microbiome, which I believe is a really wonderful system to study natural populations because you can actually capture ecological and evolutionary forces acting almost simultaneously, something that likely is generic to many organisms. And so today I'm going to take you through a couple of stories, and first I will start on Drosophila, and then I will share with you work on the human microbiome. So before I start on that story, I wanted to first tell you about what I am particularly fascinated about when it comes to evolution in natural populations, and that is how rapid is evolution? So this is an age-old question, and in the 1800s, Charles Darwin, he thought that evolution by natural selection is a slow and gradual process. Species evolve through the accumulation of small variations over long periods of time. So Darwin thought that evolution can be very slow. But then if we take a look at contemporary examples of evolution, we can see that it's actually very rapid. So, for example, the application of pesticides time and again has resulted in resistance evolving within a mere decade. And we see the same story again with antibiotic resistance. And so the question is evolution slow, like Darwin thought, or is it rapid, like we're seeing with these examples? And the answers to these questions may become apparent through querying new genomic data with new statistical methods. Now we are in an era of whole genome sequences and abundant data, and so we can start to actually probe these age-old questions in new ways. And so that takes me back to me and what I've been working on. And so first I'm going to start on the story on rapid adaptation in fruit flies. And so why Drosophila melanogaster? It's actually a really wonderful system to study because we have so much knowledge of its biology and its life history. And we also, moreover, have a lot of data for this organism. So we have almost 200 fully sequenced inbred lines from North Carolina. We have another 200 from Zambia. We have several hundred from Europe. Combined, this makes a really unparalleled data set to probe evolutionary questions. And so, this is in contrast to what was historically available, where we used to have little fragments that were just, 20 to 50 base pairs long, scattered throughout the genome. And now we actually have this whole genomic data that can fill in the blanks. And now we can actually look at the correlations between sites to make inferences about evolution. So, what kind of signatures of selection should we expect from such whole genomic data when evolution is slow versus rapid? So let's consider when evolution is really slow. You might have a very long waiting time until a new mutation arises, and here it is on the background of a haplotype. And if it survives drift, it will go to high frequency and drag with it the linked genomic region to high frequency, and that results in a hard selective sweep. Over time, that signature will decay, but importantly, in the region immediately surrounding the adapted mutation, you have one haplotype at high frequency. Now, let's consider the case where you have very rapid adaptation. In this case, you have very large census size or a large input mutation rate or a lot of standing genetic variation. Here, you'll have a very short waiting time until a new mutation arises, and it will be so short that a second one will arise on the background of a different haplotype. And then, if both survive drift, you will have both rising to high frequency, and this will result in a signature of a soft sweep where you have two or more haplotypes instead of just one. And so what I've shown you are signatures of selection associated with slow and gradual adaptation versus rapid adaptation. And now the question is, what kind of signatures should we see in Drosophila data? If we see soft sweeps, then we can conclude that adaptation can actually be very rapid. So before we start to look for such signatures in the data, we can start to think about what are the signatures associated with known adaptations in Drosophila. And so there are three very prominent cases of adaptation at the loci CYP61, Chekhov1, and ACE. And these are all cases that have arisen due to very strong selective pressures and recent selective pressures. And if we take this data set from North Carolina, almost 200 inbred lines known as the Drosophila Genetic Reference Panel, and we draw a 10 kilobase pair window around these, centered at these three loci, and then plot what the haplotype frequency spectra are, we can see that we actually have signatures of soft sweeps. So specifically, what I'm showing is that here is a haplotype present in about 50 individuals, another in about 20, and 20, and so on, and these are all singletons. And so basically, we have multiple haplotypes at high frequency, which is a hallmark of soft sweeps. But actually, we don't even need to conclude this from genomic signatures in the DGRP data. These are actually known soft sweeps. In the case of ACE, we've seen multiple independent mutations arising in different parts of the globe and even in the same location. In the case of Chekhov1, we know that we have a case of soft sweeps arising from standing genetic variation from a transposon element. And in the case of CYP61, again, we have multiple de novo mutations, but from transposon elements. So these are really well characterized and well studied in the literature. Now, importantly, they were not originally discovered via genomic scans, and instead were discovered by in-depth PopGen analyses and experimental studies. And so the question is, how many more soft sweeps are there? And crucially, at the time when I started this work, methods for detecting selection were expressly developed for looking for hard sweeps, not for soft sweeps. So we wanted to know if there could be a method that could detect soft sweeps with greater power. And so that led us to these questions of how many hard and soft sweeps are there, and can we develop a method to detect and distinguish hard and soft sweeps? So this brings me to the method that we developed, where we used haplotype homozygosity defined in long analysis windows to identify sweeps. And so here's a roughly 10 kb window, just like I showed you previously. And I'm looking at the haplotype frequencies. Here's the first haplotype. It has a frequency of T1 and so on. And we can compute homozygosity as the sum of the squares of the frequencies in this window. And so here's the first haplotype squared and so on. And so the colored area of this box is roughly equal to the homozygosity of this window. Now, all things being equal, you can see that the soft sweep has a roughly greater colored area than the hard sweep. And so homozygosity will be more sensitive for detecting hard sweeps than soft sweeps. Yet we wanted to have a statistic that could detect both with more equal power. And so that brings us to the statistic we developed called H12, where we combined the first and the second most haplotype in this common haplotype, single haplotype, and then we took the square. And what this does is for a hard sweep, it only augments homozygosity just a little bit, but for a soft sweep, it actually adds a lot. And this through extensive tests in simulations where we varied the partial frequency, the selective pressure, and the age of the sweep, we were able to see that H12 has very good power for detecting recent and strong selective sweeps, and especially hard and soft sweeps, both of them together. And so we then applied this statistic to the Drosophila Genetic Reference Panel, and here are the results of that scan. Here are the 4 autosomal arms. On the y-axis, I have H12. On the x-axis, I have the position. And then each dot represents H12 measured in a approximately 10 kb window, or about 401 SNPs. What we see at first glance is that there's a lot of structure in the data. So we first excluded regions with low recombination, but that left a lot of structure still. And so we wanted to know what else could be generating this structure. Well, it turns out that the three most prominent peaks are our three positive controls, which was very exciting for us because it meant that our method could detect soft sweeps, but moreover, it could detect soft sweeps arising from standing genetic variation and also de novo mutations. What about the rest of these peaks? So first, we wanted to rule out the fact that demography could play a role here. And so the state-of-the-art model for North American Drosophila is that of an admixture model in which there was an admixture event between an African and a European population. And we tested whether this model could generate peaks of homozygosity observed in the data. Moreover, we tested about 72 total models in which we tested simple models with different strengths of bottlenecks or population sizes, and many variations of admixture models that include different population sizes, admixture levels, migration, expansion rates, and so on. And what we found is that none of these demographic models actually fit the model especially well in regards to LD, short and long range LD, SFS statistics, including pi and S. But there are some models that fit the data much better than others. And when we consider those models, we find that actually they cannot generate these outlier peaks. We simulated under all of these models extensively, and some of them are outrageously bad. Some of them are pretty good for several of the statistics, but not all. So what this means is that there's actually a lot of work still left in terms of identifying a well-fitting demographic model. But in the meanwhile, if we consider the range of possibilities that do actually fit the parameter space reasonably well, we can start to understand whether or not homozygosity by chance can be elevated. And what we find is that under those well-fitting models, we actually cannot generate these peaks of homozygosity. And moreover, by examining these outliers, we can start to focus on the most extreme events that are more likely to be under selection. And finally, it's true that these three positive controls were actually under strong positive selection. And so the fact that they have high homozygosity is also very reassuring. So then we identified our top 50 outliers and asked whether or not these could be generated under hard or soft sweeps. We wanted to know, what are they? So we then plotted the haplotype frequency spectra for the top 50 peaks. Here are the top outliers, top 10 outliers in the data, and you've actually already seen the first three. And you can see actually for all of them, they look soft. There are multiple haplotypes at high frequency. Now we're just eyeballing the data here, and so we wanted to come up with a statistic that could actually distinguish whether these are hard or soft using another statistic called H2 over H1. What H2 is its homozygosity computed excluding the most common haplotype. And so for a hard sweep, this should result in a very small homozygosity because you're excluding the main contributor to homozygosity. But for a soft sweep, we see that homozygosity should still be somewhat elevated because you have at least one main contributor left. If we take the ratio of H2 over H1, we can augment our power to distinguish hard and soft sweeps, and it should be small for a hard sweep and large for a soft sweep. We found that H2 over H1 has an inverse correlation with H12, and this means that actually when we use H2 over H1, we need to condition on values of H12 being A, higher than what we'd expect under neutrality, otherwise there's no evidence for a sweep in the first place. And B, it should actually be matching this H12 value because of this inverse correlation. And when we plot the top 50 points, you can see it roughly follows this shape. Here are three outliers, 3 positive controls. And we wanted to know now whether these values could actually be generated under hard versus soft sweeps. And so we simulated hard sweeps by drawing the partial frequency, age, and selective strength from uniform priors. And what we plotted does not actually overlap our observed data distribution. We did the same under soft sweeps, and now it does overlap. And we repeated this actually under several demographic models, and we get very similar results. And so our conclusion, using a Bayes factor analysis, is that there's strong support for 50 out of 50 cases for soft sweeps and decisive support for soft sweeps for 44 out of 50 cases. So what we showed is that soft sweeps are actually common in Drosophila and melanogaster, far more common than what was previously thought. And this was actually really exciting news. It meant that actually adaptation can be really rapid. And it also opened up the possibility of being able to look for them more generically in other organisms. And indeed, I've been very happy to see that these statistics have been applied to many organisms, including soybeans and humans and pigs, stickleback, malaria, and more. And recently, we developed an unphased version called G12 and G2 and this is capable of expanding our space towards dealing with non-model organisms, and we recently applied it to New York City rat data and found several interesting candidates of selection. And so this has been a really exciting area of work for me, and we continue to develop these statistics for new applications and also for making these more widely applicable for people. Now, it is my pleasure to next move on to my microbiome work, but before I do that, I thought I'd pause for a moment and ask if anybody has a question or two. Be happy to take them. And if not, that's okay. I will have time at the end for more questions. Okay, so I finished this work on Drosophila, and I was really excited about rapid adaptation. But I really wanted to know, how does adaptation impact our health? And when I say that, I mean on timescales that are relevant for our own lives. And I thought, what better place to look than the human microbiome, because we have this melange of bacteria and viruses, archaea, and fungi that are inside of us that are rapidly growing and have a great potential to evolve. And we know that the microbiome has a really important impact on our health, but yet we have very little understanding of the genetics of the microbes inside of us. But I'll also argue that from a very fundamental science perspective, this is actually a wonderful system to study in terms of the complexity of natural populations. So specifically, we have potential for rapid evolution in the context of a very, very rich ecosystem where we have hundreds of interacting species. And this is actually what is generically happening, I think, for many organisms, except we study them in isolation. In this case, bacteria have short generation time, so we might be able to witness their evolution rapidly. We also have this really unique situation where we have hundreds and thousands of replicates walking around on this planet Earth ready to be sampled. And so we have like this very unique replicate ecosystem that we can keep sampling. Whereas for Drosophila, we have to go out all the way to one orchard and another, and it's less accessible for us. And then finally, of course, the added bonus is that we may actually have relevance to our health. So what do we know about rapid evolution of microbes? Well, we can take some lessons from the lab, and several scientists have been studying rapid evolution of bacteria in test tubes. Take Richard Lenski's famous experiments where he studied E. coli evolving in the lab. What he and his colleagues found are that bacteria evolve rapidly and repeatedly over time. Now, this was done in simple communities of one species studied at a time. But in the microbiome, we have a very complex community, and it's unclear whether this picture of rapid adaptation in a simple community translates to that of a complex community. Instead of seeing evolution, we might actually see species fluctuation, where you have this change in species compositions over time. Alternatively, in response to a selective pressure, you might see invasions, where a better fit strain enters from the outside environment. Or you might actually have evolution, where you have a genetic change that then propagates through its subpopulation. And when I say evolution, what I mean is a genetic change that subsequent genetic changes can accumulate on top of, not species fluctuations, not strain fluctuations. And there's actually a lot of potential for evolution in our microbiome, given the very high mutation rate and census population sizes. And in fact, Zhao and Lieberman have estimated that almost 1 billion mutations enter our microbiomes daily, making this a true hotbed or really having a very, very high potential for rapid evolution. So we've just outlined 3 possible scenarios by which the microbiome can respond to selective pressure. And right now, I'm going to explain to you what we found, which is a collaboration with Benjamin Good, which was recently published in PLOS Biology. So the overview of this part of the talk is how do we track evolution within the microbiome. Then I will share with you what evolutionary dynamics on short timescales looks like, and then evolutionary dynamics over our lifetime. So traditionally, people have been studying the microbiome using 16S data, or amplicon data, which barcodes which species are present. This is a really affordable technology and has yielded a lot of insights on the ecological species dynamics in the microbiome. But we miss a lot of information on the genetic information around in the rest of the genome. And so this makes it difficult to study evolution because you're not actually measuring the genomic variants that are relevant. So we can instead use a different technology, such as whole genome shotgun sequencing, to sample the rest of the genome. And what this does is it takes all the DNA present in a sample and shears it and sequences it. And so this is really exciting because now you're sampling the entire ecosystem, the entire genome, but it's also really challenging data because these are very short fragments of just 100 base pairs, and somehow we need to make evolutionary inferences from this. And this is a really far cry, I would argue, from the nice haplotype data that we were dealing with for the Drosophila data, where we have these really, really lovely, long-spaced haplotypes, whole genome. Somehow we have to go from this jigsaw puzzle to that, or something that looks like that, to be able to make some evolutionary inferences. And ideally, to make evolutionary inferences, what we would do is look for mutations that go from low frequency to high frequency over time. And how do we do that? So first, what we want to do is try to simplify this data. And so what we can do is try to measure genetic variation within species. And so we can map reads to different reference genomes and then measure allele frequencies within each reference genome one at a time. And so, for example, at this site, we have an allele present at 50%. So similarly, we could take a look at the blue species, for example, and we can see across a panel of individuals that this particular site has an allele frequency at 0.8, 0.3, 0, etc. It's actually a very variable site in this cartoon, suggesting it might be a very interesting candidate to look at. Okay, so we want to look for these allele frequency changes going from low to high after simplifying the data. But that's not that simple. So what could actually be generating these allele frequency changes instead of evolution could be strain fluctuations. So multiple strains of the same species often colonize a host, and these strains are distinct strains because they have genetically diverged from each other. And so these divergences manifest themselves as polymorphisms inside of the host. But we don't actually witness this population structure inside the host. All we witness are actually just allele frequencies. And so somehow we need to go from these allele frequencies to inferring what the population structure is so that we can disentangle strain fluctuations from evolution. So how are we going to do that? So here, what I'm doing is I'm actually taking these allele frequencies and I'm going to plot them as a histogram. And so here I'm plotting histograms for 3 individuals from the Human Microbiome Project for the common species Bacteroides vulgaris. And on the x-axis, I have the allele frequency. On the y-axis, I have the fraction of sites. And so what you can see is that we have a lot of variation across hosts. We see these very funny bumps that to a population geneticist may feel non-standard because we don't expect to see these intermediate frequency bumps. And we can try to start to understand these distributions to infer the lineage structure inside of hosts. And so here's actually a cartoon of what's likely happening inside of hosts. So let's walk through this. In this first host, we have a bump or a mode at around 15%. And so what's happening is that this person is likely colonized by a strain at 15% and 85% respectively. And the evolutionary differences that accumulated before colonizing the host are what are manifesting themselves as these polymorphisms out here, which are present at the relative frequencies of these 2 strains. So I say that there's one strain at high frequency because there's basically one common strain, and then there's one subdominant strain. Here we have a very similar situation where we have a mode at roughly 50%, and so we have roughly 2 strains present at intermediate frequency, 50-50 frequency. And here we have a situation where we have multiple modes, and likely there are 3 or 4 or some other discrete number of strains at high frequency, but we can't actually parse what exactly is the lineage structure here. And so just by looking at these patterns, one of the conclusions that we can make already about the lineage structure inside of us for a given species is that often we're colonized by a handful of strains, 1, 2, maybe 4, but not hundreds and not exactly 1. Now, for other species, we see something very similar. For some species, we do actually see exactly 1. But we rarely or never have observed actually more than a handful. And this question of oligocolonization, how it happens, why it happens, is something very interesting that we can investigate in future research, but for now we leave as a point of interesting discussion. And the nice thing is that we can actually leverage this lineage structure to parse the lineage structure inside of the host, or more concretely, to phase the haplotypes of the dominant lineages when possible, so that we can track evolution over time. So some of these samples, as I mentioned, are too complicated, but other samples we can work with. So here's a situation where we can actually phase the dominant haplotype so that we can track evolution over time. If we take a look at 2 sites, we have a pileup of reads, and the majority will come from the red strain, and the minority will come from the blue strain. And so we can assign the majority allele to the majority strain, and in that manner, we can quasi-phase, or in some sense, phase with high confidence the majority of the alleles on the background of this lineage. That's not so easy for this sample because, as you can see, there's a 50-50 chance of assigning the wrong allele to the wrong strain. And so, therefore, any allele frequency changes could be confounded with actually phasing errors. So we want to focus on those samples in which we can actually phase the dominant lineage, and then we can track evolution on the background of that dominant lineage. And so you might be asking, how many such samples do we actually have that meet this criteria? And highlighted in dark blue are those such samples for the top 30-ish species that are prevalent in our microbiomes. And you can see that actually there's a lot of data. combined across the top 30 species, we actually have 801 resident populations across hosts and species that we can work with. And so this is actually really wonderful because it really opens the door in future work for very nice comparative population genetics work as well. Okay, so we've done all this hard work for phasing the dominant lineage. Now we're in the position to track evolutionary dynamics on short timescales. So, do bacteria evolve in the host gut within just a few months? We took data from the Human Microbiome Project, and we looked for these kinds of signatures where we saw an allele going from low to high frequency on the background of a dominant lineage. And what we're going to plot now are the results of those computations, where on the x-axis I have the number of SNP changes, and on the y-axis I have the fraction of comparisons with n or more SNP changes. And here's what the data looks like. So first, what we have actually is, because it's plotted on a log scale, 0 changes acting in 88% of our populations. So not much seems to be happening actually on short timescales, which is not something new, but what we did notice is that a few of these populations, 9%, have a few changes on the order of 5 or 10 SNP changes. And a lot of changes happen in just a small handful of just 3% of our samples. So we have the majority of our samples having 0 changes, 9% having few changes, and 3% having a lot. It turns out that 2 processes are likely generating this distribution. It's this funny bimodal distribution where either you just have 5 or 10 or 10,000 SNP changes. What could those 2 processes be? Well, it turns out that the 2 processes are invasion versus evolution. Let's think about that. So if we take our human microbiome host and we take the B. vulgaris bacterium, for example, and we make a tree, we can see the evolutionary history of all the strains of B. vulgaris collected across these different hosts. And B. vulgaris, over a long evolutionary time, would have accumulated a lot of changes. Whereas over a little bit of time, we would expect to see, for evolution, just a small handful of changes accumulating. For an invasion, we'd see that lots of changes would enter the host due to a lot of evolutionary time having passed. So basically, we can distinguish what's going on within hosts versus between hosts. We can distinguish evolution from invasion by comparing what's going on within hosts with what's going on between hosts. We can assume that there's long evolutionary distances separating B. vulgaris strains from between hosts, and that actually does indeed result in a mean number of 10,000 SNP differences between hosts, whereas within hosts we have on the order of about 100 SNP changes that differ between on 6-month timescale. And that is actually averaging over what's going on here and here. And if we plot on this plot the number of SNP differences between hosts, you can see that on average we have about 10,000 SNP changes. That allows us to categorize what's going on within hosts as either evolution or invasion, and because the invasions are more consistent with what's going on between hosts. So this is actually really exciting. For the first time with our machinery, our ability to phase the dominant haplotype, we were able to actually see in this very complex and messy data evolution. And this had not really been observed before. It had not been noted that actually commensal bacteria inside of us can evolve so rapidly. Simultaneously, Zhao and Lieberman made a similar discovery with B. fragilis using isolate data. And so independently, we were able to show that, yes, indeed, the commensal bacteria inside of us can evolve. And this suggested a lot of potential for our microbiomes to change dynamically on short timescales and actually impart potentially phenotypic changes also to the host. But this is just the start. We are just discovering that these commensal bacteria can evolve. And now we can ask, well, how much evolution is there? What is under selection? Are there limitations to the amount of evolution in a host? And how much evolution, or how does evolution happen? These and many other questions remain. I'll try to take a small stab at a few of these right now in the time remaining. So one of the questions we wanted to ask was, does the broader community play a role in the rapid adaptation? We are, after all, studying these evolutionary changes in the context of this complex community. And to investigate that, we asked, well, are de novo mutations more commonly the mechanism of evolution, or is it via adaptive introgression or recombination-seeded sweeps? When I say adaptive introgression, I mean you have a donor that gives a chunk of its DNA to the recipient and then swaps out that gene. There could be gene gains and losses and a chunk of DNA swapping out. And this scenario actually makes sense in the microbiome, given that you have this huge melting pot of a reservoir of pre-adapted genetic material. And in the face of new changing environments, recombination-seeded sweep could actually be more efficient than evolving a de novo adaptation. And indeed, we actually saw a lot of evidence for adaptive introgression, one being that we saw a lot of gene gains and losses, but also because we saw overall the mean DNDS of SNPs changing within hosts was actually far less than 1. So that actually suggested a very important role for recombination in the microbiome and for the broader community in seeding adaptive sweeps. Now, the next question we wanted to ask was, what are the evolutionary dynamics over our lifetimes? We've just looked at what's going on inside of us on 6-month timescales, but do these bacteria continue to evolve as we grow old? We saw that a small percentage of our strains get replaced after 6 months. Does this strain replacement actually continue after throughout our lifetime, such that every strain in our body gets replaced after 20 years? Or are some hosts extra susceptible? Maybe because they consumed antibiotics or traveled or had an illness. Maybe the replacements are specific to those hosts with special conditions. Or maybe some strains become so well adapted that replacements become less likely with age. They continue to evolve such that they become so specialized to us that no other strain can enter. And so we wanted to ask these different questions, to ask, how does the long-term evolutionary dynamics play out in our microbiomes throughout our lifetime? So the ideal data for this is to sample longitudinally over a few decades. But as you know, this is actually very difficult to do. And so instead, what we did was we studied twins. So it turns out that there's a study by Corpella et al. That shows that cohabiting teenage twins and siblings share many strains. And we found a data set by Shi et al. In which 125 adult twin pairs had grown up together in the same household and then lived for 40 years apart. And we asked the question of whether adult twins share the same strain. If they do, then it means the strains inside of us continue to evolve and stick with us for our lifetimes. But if they don't, that means that the strains inside of us get lost and are completely replaced. So we're going to add yet another line on this plot, and here it is. This is for the between twin SNP changes. So on average, there are about 10,000 SNP changes between twins for pretty much any given bacteria species strain pair. And so what this means actually is that replacements are very common over our lifetimes. These high number of SNP changes is too great to accumulate on just 6-month timescales. On 40-year timescales. On 40-year timescales, you would expect maybe at most 800 or 1,000 SNP changes. But actually what we're observing is 10,000 SNP changes. And so this suggests that while on short timescales we see a lot of evolution, on long timescales replacements dominate. Why do we see these replacements? And do replacements gradually happen or in bursts? We don't know. We don't know the answers to these questions. But these are exciting avenues for future research. So to conclude this part of the talk, we can say a few things about the evolutionary dynamics of bacteria in the human gut. Hosts are oligo-colonized by just a handful of strains per species. On 6-month timescales, bacteria can evolve in the human microbiome. Replacements are rare. Over our lifetimes, bacteria are replaced. This suggests limits to how well adapted bacteria become to us. And the broader community of bacteria play a critical role in providing the material for recombination of seeded sweets. And more broadly, going back to my broader mission, which is to study evolution in natural populations, it seems that rapid adaptation and soft selective sweeps are common in a wide range of organisms. And our current understanding of the tempo and mode of adaptation will need to be revisited given new genomic data and the community complexity that organisms experience. And so I do believe that we are still very much at the beginning of this journey, especially with regards to the human microbiome, where we're just starting to see that there's evolution happening on such short timescales, but there's just so much more to understand. I'd like to acknowledge my co-authors and mentors, Dimitri Petrov, Philip Messer, Noah Rosenberg, and Erkan Buzbus on the Drosophila melanogaster work and soft sweets work, Benjamin Good, Katie Pollard, and Oscar Halachek on the microbiome evolution work. And this is my lab, and we're really having a lot of fun despite the quarantine and the pandemic. We're recruiting, so please get in touch. And I'm happy to take any questions. And thank you again for this opportunity.